Hi everyone, welcome to all. This is Vidyam Academy's most expected question series for medical NEET examination. Today's topic is 12th Biology, chapter number 5, Molecular Basis of Inheritance. And into this subtopic is Transcription Genetic Code. And now it's being presented by Rajiv Patel. Once again, let's welcome to all. Let's get started. The first question is, in transcription, which strand of the DNA double helix serves as the template and in which direction is the RNA strand synthesized? Option A. The template antisense strand is a read 3 to 5 by RNA polymerase, producing RNA in the 5 to 3 direction. Option B. The coding sense strand is read 5 to 3 producing RNA in the 3 to 5 direction. Option C. Both strands are read simultaneously. 2 complementary RNA copies are made at once. Option D. The template strand is read 5 to 3 and RNA is synthesized in the 3 to 5 direction. What is the right answer? Do you guess the right answer? The right answer is option A. Let's see how and why. Go to detailed explanation. Transcription uses only one of the 2 DNA strands, the template antisense strand, as the guide for RNA synthesis. RNA polymerase reads the template strand in the 3 to 5 direction and synthesizes the new RNA strand in the 5 to 3 direction, adding ribonucleotides complementary to the template, A to U, TA, and G to C, and C to G. The other strand, the coding sense strand, has the same sequence as the RNA with U instead of T and is not read. The choice of which strand is the template varies from gene to gene. Some genes use one strand, so neighboring genes may use the opposite strand, depending on which strand carries the promoter in the correct orientation. That's what the template antisense strand is read to 3 to 5 by RNA polymerase, producing RNA in the 5 to 3 direction. Option A is the right answer, exactly what question demands for. Next, move to question number 2. In prokaryotes, RNA polymerase consists of core enzyme and sigma factor. What is the specific role of the sigma factor? Option A. Sigma factor synthesizes the RNA strand by adding ribonucleotides to the 3OH end of the growing chain. Option B. Sigma factor unwinds the DNA double helix throughout the entire gene before RNA polymerase begins synthesis. Option C. Sigma factor recognizes and binds to the promoter sequence on DNA, enabling transcription to initiate at the correct site. Option D. Sigma factor terminates transcription by recognizing the terminator sequence and disassociating the transcript. What is the right answer? Would you guess the right answer? Your time starts now. Yes, the answer is option C. Let's go to detailed explanation why option C is right answer here. The complete RNA polymerase holoenzyme in prokaryotes is core enzyme plus sigma factor. The core enzyme alone can synthesize RNA but cannot locate the correct starting position on the DNA. The sigma factor specifically recognizes the promoter, the DNA sequence upstream of the gene, by binding to 2 conserved elements. The 10 element, example, Pribnow, box, and the 35 element, this promoter recognized positions the holoenzyme precisely at the transcription start site, exactly +1. After initiation begins, the sigma factor disassociates from the complex and the core enzyme alone elongates the RNA chain. Sigma factor is therefore a specifically factor for initiation only. That's what sigma factor recognizes and binds the promoter sequence on DNA, enabling transcription to initiate at the correct site. Is option C is the right answer for this question. Question number 3. The genetic code uses a triplet codons. How many total codons are possible and how are they distributed between sense codons and stop codons? Option A, 20 total codons, one for each of the 20 amino acids with no stop codons. Option B, 64 total codons, all 64 specify amino acids, making the code non-degenerate. Option C, 16 total codons, 4 bases taken 2 at a time with 4 stop codons. Option D, 64 total codons, 61 code for amino acids, sense codons and 3 are stop codons, UAA, UAG and UGA properly. Would you guess the right answer? Yes, let's check whether you guess right or wrong. The option D is the right answer for this question. Let's see in the detailed explanation. With 4 different bases A, U, G, C and a triplet codon, the number of possible combinations is 4:3 is equal to 64 codons. Of these, 61 are sense codons, each specifying one of the 20 standard amino acids. The remaining 3 are stop nonsense termination codons, UAA, ochre, UAG, amber, and UGA, opal. So they do not code for any amino acid, but signal the ribosome to end translation. Since 61 codons serve only 20 amino acids, most amino acids are encoded by more than one codon. This is called degeneracy. For example, leucine has 6 codons and serine has 6 codons, with methionine and tryptophan are each encoded by just one codon, AUG and UGG. That's what option D is the right answer for this question. Next, question number 4. Which of the following correctly identifies the start codon and 3 stop codons of the genetic code? Option A, start codon UGA, stop codons UAA, UAG and AUG. Option B, start codon AUG methionine, a stop codons UAA ochre, UAG amber, UGA opal. Option C, start codon UAA, stop codons AUG, UGA and UGG. Option D, start codon GUG, stop codons UAA, UAG, UAU. What is the right answer for this question? Yes, let's check whether your guess is right or wrong. Option B is right answer. Let's see why and how. AUG is the universal start codon. It initiates translation in virtually all organisms and codes for methionine. In prokaryotes, the interior methionine is formulated. The 3 stop codons UAA, UAG and UGA signal the end of translation. No tRNA recognizes these codons. Instead, protein releases factor bind and cause the ribosome to release the completed polypeptide. UA is called ochre, UA is amber, UGA is opal, umber, UGG, tryptophan, and UGA, stop are easy to confuse. Only UGG codes for an amino acids. UGA is always a stop codon in standard genetic code. Knowing all 4, AUG start, UAA, UAG, UGA. That's what option B is right answer for this question. Next, move to question number 5. Which of the following correctly describes the 2 properties of the genetic code? Option A, universality. Some codons specify same amino acids in nearly all organisms. So degeneracy, most amino acids are encoded by more than one codon. Option B, universality, all 64 codons are used equally in all organisms. Degeneracy, each codon codes for more than one amino acid. Option C, universality, the code works only in prokaryotes. Degeneracy, the number of codons exactly equals the number of amino acids. Option D, universality, the code is specific and varies greatly between organisms. Degeneracy, only stop codons or degenerate. What is the right answer for this question? Which of the following correctly describe 2 properties of the genetic code? Let's check. What is the right answer? Option A is right answer for this question. Let's see why and how. Universality means the genetic code is essentially the same across all life forms. AUG codes for methionine in bacteria, plants and humans alike. This conversation suggests the code was fixed very early in evolution. Minor exception exists. Mitochondria and some protists use slightly variant codes. Degeneracy means most amino acids are specified by more than one codon. 61 codons, so 20 amino acids. So on average, each amino acid has about 3 synonymous codons. Degeneracy is not ambiguity. So one codon always specifies the same amino acid, non-ambiguous, but one amino acid can be specified by multiple codons. Synonymous codons typically differ only at the third obal position, which is why many third position mutations are silent. They do not change the amino acid. That's what universality, same codon specifies same amino acids in nearly all organisms. Degeneracy, most amino acids are encoded by more than one codon is exactly right answer for this question. Next, move to question number 6. In eukaryotes, the primary transcript, pre-mRNA, undergoes 3 major processing steps in the nucleus before becoming functional mRNA. Option A, capping plus translation plus tailing. mRNA is directly translated after cap and tail are added. Option B, methylation, phosphorylation, and glycolization. Chemical base modifications improve stability. Option C, 5M7G capping plus 3 poly A tailing and splicing intron removal by splicem, producing mature mRNA for export. Option D, splicing plus editing, silencing, 3 steps that reduce mRNA length and activity. So what is the correct answer for this question? Option C is exactly right answer for this question. Let's see why and how. Eukaryote pre-mRNA must be processed before it can function. 3 modifications occur in the nucleus. 5 capping, A7 methylguanine cap is added to the 5 end. It protects the mRNA from 5 exonuclease degradation and is essential for ribosome recognition during translation initiation. So 3 poly A tailing, approximately 200 adenine residues are added to the 3N by poly A polymerase after cleavage at the polymerization signal. This tail enhances mRNA stability and aids nucleus export. Spliceosome complex precisely removes non-coding introns and joins the coding exons together. After all 3 modifications, the mature mRNA is exported through nuclear pores into the cytoplasm for translation. That's what option C is exactly right answer for the question which 6 demands for. Next, question number 7. Francis Crick's obal hypothesis explains why fewer than 61 tRNA types are needed to decode all 61 sense codons. What does the obal hypothesis state? Option A, the first 2 positions of the codon are flexible and can pair with any anticodon base. Option B, all 3 positions of the codon pair strictly with the anticodon. Exactly 61 tRNAs are required. Option C, the obal position is the first base of the codon. One codon can specify multiple amino acids. Option D, the third codon position, obal position, 3 end of codon, allows non-standard base pairing with the anticodon's first base. One tRNA can read multiple synonymous codons. What is the right answer? Yes, option D is right answer. Let's see why and how. The obal hypothesis, Crick 1966, explained why cells can decode all 61 sense codons using fewer than 61 different tRNA molecules. The first 2 codon positions pair strictly with the anticodon by standard Watson-Crick rules. However, the third codon position, the obal position, 3 end of codon, can form non-standard base pairs with the first anticodon position, 5 end of anticodon. For example, inosine, a modified base in many anticodons, can pair with the UCA at the obal position. This means a single tRNA with one at its anticodon position can read 3 different codons that differ only at the third position. Since synonymous codons typically differ at the third position, obal pairing allows one tRNA to serve multiple codons encoding the same amino acid, reducing the total number of a distinct tRNA required. That's what option D is exact right answer for this question. Question number 8, match the column. 1 is a term. So with the column 2, correct description. The first one, promoter. Option A, 3 base sequence to tRNA brings correct amino acid to ribosome. So here left hand side 2 codon, B DNA sequence that is transcribed into a functional product. 3 anticodon, C DNA sequence upstream of gene, RNA polymerase binding and initiation site. 4 structural gene. So here D 3 consecutive nucleotides in mRNA specifies 1 amino acid. So here are the options. Option A 1A 2D 3B 4C. Option B 1B 2C 3D 4A. Option C 1C 2D 3A 4B. Option D 1D 2A 3C 4B. What is the right answer? Yes, the absolute right answer is option C. Let's check how and why. Promoter 1C, a specific DNA sequence upstream of the transcription start site where RNA polymerase binds to initiate transcription. In E. coli, it contains the 10 box and 35 elements recognized by sigma factor. So codon 2. A triplet consecutive nucleotides in the mRNA that the ribosome reads. Each codon specifies 1 amino acid or a stop signal. Anticodon is 3, a 3-base a sequence located in the anticodon loop of the tRNA molecule, complementary and antiparallel to the mRNA codon. It ensures the correct amino acid is delivered to the ribosome during translation. So structural gene, it's a 4-1B. Any DNA segment that is transcribed to produce a functional product, whether mRNA encoding a protein, tRNA, or rRNA. That's what option C, which matches correctly, is the right answer. Next, move to question number 9. So here assertion A, in prokaryotes, transcription and translation can occur simultaneously. Ribosomes begin translating the mRNA while it's still being transcribed. So reason or, prokaryotes lack a nucleus. Transcription occurs in the cytoplasm, so where ribosomes are present. So a ribosome can bind the 5 end of the mRNA as it emerges from RNA polymerase before transcription is complete. So option A, both A and R are true and R is the not correct explanation of A. Option B, both A and R are true but R is not the correct explanation of A. Option C, A is true but R is false. Option D, A is false. Translation only begins after transcription is fully complete in prokaryotes. Would you guess the right answer? The correct answer is option A. Both A and R are true and R is the correct explanation of A. Let's see. Simultaneous transcription and translation, called coupled transcription translation, is defining a feature of genes expression and has been directly visualized by electron microscopy showing a polysome. So multi-ribosomes attached to mRNA still linked to RNA polymerase and the DNA. Reason R also true and gives the structural basis. Prokaryotes have no nuclear membrane separating the genome from the cytoplasm. All processes occur in the one compartment. As soon as the 5 end of the mRNA is synthesized and carries the chain ribosome binding sequence, a ribosome can bind and begin translation immediately. Or directly and casually explains the absence of nuclear membrane or is the structural reason that simultaneous transcription translation A is possible. In eukaryotes, transcription occurs inside the nucleus and the translation of the cytoplasm, they are spatially and temporally separated. And final question of this topic, question number 10. Arrange the following events in correct order from DNA to mature mRNA eukaryotic cell. P. RNA polymerase 2 binds the promoter, transcription begins. Q. Primary transcript pre-mRNA synthesized contains both exon and introns. R. 7-methylguanosine cap added to the 5 end. Yes. Polyetyl 200 adenine residues added to the 3 end. T. Spliceosome removes introns, exons join to form continuous coding sequence. U. Mature mRNA exported through nuclear pores into the cytoplasm. Option A. QPRTSU. Option B. PQTRSU. C. PQRSTU. And option final D is RPQSTU. What is the right answer? Option C. PQRSTU is right answer. Let's check how and why. The sequence begins with pRNA polymerase 2 guided by transcription factors binds the promoter. So here 25 in eukaryotes and initiate transcription. So Q follows the entire primary transcript. So pre-mRNA containing all exon and intron sequences. So E is synthesized or happens co-transcriptionally. The 5M7G cap is added to the 5 end almost immediately as the first 20 to 30 nucleotides are synthesized. While the polymerase is still transcribing, the rest of the gene S occurs after transcription terminates. The poly A polymerase adds 200 adenine residues to the 3 end following cleavage at the AAU-AAA polyadenylation signal. T follows the spliceosome assembles and the capate polyadenylated pre-mRNA and sequentially removes each intron, precisely joining adjacent exons. Finally, U, the fully processed mature mRNA, is recognized by export factors and transported through nuclear pores into the cytoplasm for ribosomal translation. That's what PQRSTU option C is the right answer. So here we explained 10 most important and expected questions for your upcoming NEET examinations. And next, I will come up with another topic which are very important for your NEET examination. Till so far, I would like to conclude this session with very well goodness. Thank you and thank you so much to all.